Okay, I think, you know, we will restart. This is a live event, and this is the joys of living through a pandemic. This is, you know, the type of things that we are learning new skill sets. So what I'm going to do is start again. So hi everyone. So I'm Professor Sarah Rankin, and I'd like to welcome you to the Imperial COVID-19 lockdown lessons. Today we've got lesson 1, and we're going to be hearing from Professor Altmann about the immune response to COVID-19. But before this, I'd like to introduce you to a couple of our students, Esther and Karim. And I know that a lot of you are going to be applying to university or college in the next couple of years. And so I want to take this opportunity to introduce you to some of the subjects that you have not studied at A-levels, but you could possibly study at uni or college. So Esther, can you start by first telling us again what immunology is?
So immunology is the study of the body's defense against infection. And so here are some key terms relating to immunology and what it is. So when we study immunology, we really study how different organs, tissues, and cells work together to mount on an immune response. And these different organs, tissues, and cells make up the immune system. And the immune system, it develops as straight away as we're born, and it plays an integral role in health and disease. So it's very important to know how we fight off these infections and how we develop immunity to microorganisms through the immune system.
Great. So why is it so important as a, you know, what's the application of immunology?
So immunology plays an integral role in health and disease, as I've said, and there's many different job titles of an immunologist. So an immunologist can study more about infectious diseases and how they evade the immune system and how the body defends itself against infectious diseases. And also in doing this, we can learn more about vaccine development and drug development. And also in cancer research, immunology is very important. And for treatment for cancer, immunotherapy has been tried out, and there's a lot of research going into this. Also understanding immune disorders, such as autoimmune diseases and immunodeficiencies. I mean, so immunologists can work in the lab, and there's also some bioinformatics that people can do on computers, processing the results. So there's quite a few different things you can do as an immunologist.
Yeah, I gather from Danny that you've got great computer skills yourself, and you did a project in his lab analysing large data sets and looking at the sort of relationship between bacterial infections and particular types of lung diseases.
Yeah, so I learned some basic computing in my undergraduate degree and also as part of workshops in my master's. But I've recently, in Danny's lab, I was able to learn how to use specific programs such as R Studio, and I found it to be something I really enjoyed. And I think that I'm quite good at it, so I quite enjoy it. And that's another avenue when you're looking at being a researcher. You can also do stuff on computer programs if you're not too fond of the lab and you don't really like the lab work.
Yeah, so I think everybody thinks of practicals as the type of practicals that we would refer to as wet practicals, which are in the lab. But actually, what you're talking about is a sort of what we would refer to as a dry lab project, something you can do in an office or now at home. But anyway, just tell me how you decided to become an immunologist, or what degree did you actually do, and what was your decision process, I guess?
So for my undergraduate degree, I did biomedical science. And I did this because it's there's loads of topics that are actually covered in biomedical science. And in the first year, you do a common year where you learn like the basics of biology. And this was very broad. But in the second year, we got to choose different modules. And what I quite liked was the microbiology and immunology module. And from this, I went on to doing a master's in virology, where I learned more about how viruses can evade the immune system and how the immune system works to fight against viruses. And so I really enjoyed this.
Great. Okay, thanks, Esther. That was really great. So, Karim, I know you're doing something slightly different, and you're doing a different type of experiment that we would refer to more as a wet lab project. So, Karim, tell us just a little bit about your backstory and the type of experiments that you do.
Okay, so I started by studying at A-level maths, biology, physics, and chemistry. And much like Esther, I didn't really know what aspect of biomedical sciences I wanted to go into. So I picked biochemistry BSc, which had a very broad common year one, and that kind of allowed me to get a flavour of the different topics. And then after that first year, we were allowed to pick our modules, and I picked immunology. And ever since then, I've always found it remarkable how the body has this it has the ability to fight almost any pathogen that's thrown at it. So I guess from there, after my undergraduate, I did a master's degree in immunology at Imperial. And during that, I was applying for PhDs, and I'm now doing a PhD in immunology at the National Heart and Lung Institute, where I'm studying respiratory immunology. So I'm looking at the airways and lungs, specifically in the context of asthma and lung fibrosis. And I'm trying to answer the question of whether we see an impaired adaptive immune response in those diseases. So whether we see impaired antibody immunity. And so what this slide actually shows is just one of the techniques that I do pretty much on a daily basis, whereby we get fresh human samples from the hospital. So patients come in with asthma and other chronic lung diseases, and we get samples. So we get lavage samples, which you can see on the top left. But we also get lung tissue samples occasionally. And I take these back to the lab, and then I break them up, and then I split them into a cellular kind of compartment. And then I use a technique called flow cytometry, which is a very commonly used immunological technique, which allows me to look at the different types of white blood cells in these tissues. And you can see there are loads of different types of white blood cells or leukocytes. So we've got our lymphocytes, we have our granulocytes, so eosinophils and basophils. But what flow cytometry allows us to do is to look at the numbers of these cells in the different tissues from healthy people and also people with disease, and see if there's any difference in those cells. And from that, we can kind of assess how severe their disease is and whether they might respond better to certain therapies.
Yeah, so first of all, Karim, just assure me, that looks like a whole lung you've got there that you've extracted from somebody. What sort of size of lung sample have you actually taken?
So that is a piece of human lung. It weighed about 8 grams. And
how
this
big is that? Big, by centimeters.
Kind of like your fist, maybe.
Whoa.
Yeah.
Okay.
And so we process that in various ways, and we look at different cells within that tissue.
Mm-hmm. Okay, and it's surprising. I think most people would be surprised. To me, that looks like a piece of liver, and it's very red. And obviously that's because it's a very vascularized organ, and you've got all these vessels, which is where you get your exchange of oxygen, yeah?
Yeah, that's correct, yeah.
Okay, cool. So, and it's interesting, this technique that Karim's showing us, because this same technique has been used to examine the white blood cells that are present in patients with COVID-19. And I think this is looking, or this leads us very nicely into Danny's talk. So thank you very much, Karim and Esther. And just to say that Karim and Esther are going to now be monitoring the chat. So if you have any questions for Karim, Esther or Danny, please put them in the chat. And if we've got time at the end, we will select out a few of those questions to answer. So like to introduce Danny, Professor Altmann. He's a world-renowned immunologist. His lab studies the immunological response to infectious diseases in a global health setting. So, for example, he has been studying immunity to the Zika virus during the Brazilian outbreak. And during this pandemic, he has been delivering international webinars for the scientific community to keep them updated on all the latest research that's coming out on COVID-19. And he's also been doing things in terms of informing our MPs. So he's been providing witness evidence to the Parliamentary Science and Technology Committee and also to the House of Lords. So we're really lucky to get such an expert in this area. Okay, so over to Danny now, and we're going to have our first lockdown lesson. So he's going to be telling us what we know so far about the immunological response to COVID-19.
Great, thank you very much. So obviously we're living in really crazy times, historic times, in so many different ways that you'll be aware of. And one of the very strange things for me, having spent my whole life in immunology, is that suddenly, you know, whoever thought we'd be in a time when world leaders are standing up and talking about PCRs and antibody tests and different kinds of antibody tests and trying to understand the differences between them. So I thought that one of the most useful things I could do would be to spend 15 minutes or so talking through some of the concepts and some of the big questions that I think really puzzle people at the moment that we're trying to grapple with. And they're certainly the questions that come up most often when journalists or news programs or whatever interview me, the things that are really bothering people. And if you get excited and suddenly think any of this stuff is good, I've put my Twitter address on the first slide, because what I tend to do is I kind of read all the research that comes out on COVID-19 immunology in real time and tweet about it many times a day. So if you know, if you think that's your cup of tea, then have a look, because I put it all up there, you know, every few hours or so. So let's just, oh, sorry. Let's get to the next slide. All right, 1 moment.
That's odd.
Well, that's better. So, you know, Esther's introduced the immune response. So the immune response is a very complicated thing. So if you were to hear about it on those Downing Street briefings, you might imagine that it was very simple and all we had to do was measure some antibodies. But you know, if you've touched on this in, you know, GCSEs or A-level courses, you'll probably be aware already that it's an enormously complex beast. So there are thousands of different types of cells that make up your immune response. They're making thousands of different types of molecules. And so viruses and bacteria getting into your body face a number of different obstacles. So at the highest possible level, we think about it as innate immunity. So as the word suggests, the kind of intrinsic barriers that anything trying to get into your body faces, whether by that we mean the cellular responses or the things like the mucosa or cilia in your nose, or the mucosa in your lungs, or the interferons. And then the next distinction that we have, oh, sorry, I'm really struggling with this, is adaptive immunity, and that's the main thing we're talking about today. So adaptive immunity, what's special about it? What we mean there is the part of the immune response that's specific for the antigens on a particular virus or bacterium. So, you know, when people talk about, you know, is this person immune to SARS-CoV-2 or not, they're saying, has this person seen this virus before and have they made a response to it? And so part of that definition is all about specificity. And part of it, I guess, is about this idea of memory and quality of response. The idea that if you've met a virus or a bacterium before, next time you meet it, you're going to be able to make a more rapid and a more potent immune response, and hopefully, therefore, it won't make you sick. And as you'll be very well aware, that's the principle that underlies vaccines. So memory is key to this, and memory is the thing that we're talking about in terms of how the population needs immunity to this virus on board if we're not going to go through the same thing all over again in a second wave or in subsequent waves. So we're talking about a response that's got special cells that have memory for the thing they've seen and specificity for the thing they've seen. So I've mentioned that there are these thousands of different types of cells, and mainly as immunologists, we're talking about white blood cells. So having kind of raised the excitement levels here about these thousands of different types of cells, if you were to take some cells from bleeding somebody from their arm and look at them down a microscope, they'd look a bit like this. So in other words, although we talk about many different cell types that certainly do have many different programs and can make many different effector molecules, down your microscope, they'd look like this. They'd look like little round blobs, not terribly exciting. And yet those different blobs you can see on the screen here can all be different types of white blood cells programmed to do very different things in the immune response. And at the most basic level, we talk about B lymphocytes and T lymphocytes, or if you like, B cells and T cells, a term that may be familiar to you. So let's try and have a look. So if we're talking about B cells, so I make no apologies for this cartoon. It comes from a series of books that I really love. And if you are at all interested in immunology, please take a look at them. It's a series of manga adult comics called Cells at Work by Akane Shimizu, who worked with immunologists at Tokyo University to really try and tell the story of the immune response. So this hero here is the B cell, and they've got a kind of squirter gun for making antibodies, which is relevant because antibodies are soluble molecules that go out into your bloodstream. And what they're doing in this case is recognizing a virus that shouldn't be there, in this case SARS-CoV-2, and trying to grab onto it before it can make it into your cells, neutralize it, and prevent you from getting infected. Okay, so that's B cells. What about T cells? This sort of mean guy here is a killer T cell, and he's got in the comic version, he's got a knife. And so, as the name suggests, what they can do is find cells where the virus has got in and recognize the viral antigens on those cells and kill those cells before any damage can occur. Okay, so at the highest level, so we had innate immunity and adaptive immunity, and with adaptive immunity, we've got these B cells that are making antibodies and these T cells that are killing infected cells. Okay, so let's do a bit of revision on what is COVID-19, this disease that's caused by SARS-CoV-2, before we look at the immune response to it. So you'll probably know it's a coronavirus that was first detected in people in Wuhan in October and November 2019. Where the first recorded published case was somebody who walked into a hospital during the first few days of November. It's a zoonosis, which obviously means an infectious disease that's transmitted to humans from animals. And these zoonoses, by which I mean coronavirus zoonoses, have been crossing over into humans for more than 1,000 years. And until recently, we tended to think of them as rather kind of benign common cold viruses. And for these zoonoses, the coronaviruses, we mainly think about getting them from bats and rodents. So I've tried to summarize that in this next slide here, which is borrowed from a review article about coronaviruses. And it simply tells the story that they've been coming into humans for a very long time. It's not a new event. But if you look at what was happening from about 1200 years ago until quite recently, there were occasional common cold viruses crossing over from the animal kingdom into humans. But the 21st century has been a really bad time for coronavirus transmissions. And we had SARS, and then we had MERS, and now we've got COVID-19. So if you were of a kind of gloomy frame of mind, you'd say, what's going on here? They're getting nastier and they're getting more common. And maybe there's a whole other discussion we could have about whether that's to do with humans and the way we're living and the way that we're impacting on the interface with animals and their habitats in the world today. So this is a virus where the sequence of it first became known on January the 11th, 2020. So, no knowledge out there whatsoever. So where on earth do you start? So one of the first things you might want to do is look at the virus and the different structures encoded by the viral genome and say, if we're trying to understand this specific adaptive immune response, which bits are targeted by the antibodies and T cells. Different parts of this incredibly multifaceted immune response could be turned on. It could be virus neutralizing antibodies. There's lots of other things that antibodies do. They coat cells in antibody to be eaten up by phagocytes. They bind complement. They bind receptors on killer cells. It could be that it's not antibody mediated at all, that we need to be thinking harder about these T cells. And T cells don't just kill, they also release lots of molecules, these cytokines, and they come in many, many different flavors. So we certainly need to understand that. And then there are many other cell types in these purple blobs that I showed you at the start of the talk, things like natural killer cells. So all of this, chapter in the textbook was blank until a few months ago. And it's been, phenomenal to see data coming out more or less on an hourly basis, and the story being kind of, filled in. So if you think about that battle between the immune system and the virus, clearly there could be different outcomes. And if you look at different people, there are different outcomes. So one outcome, the worst one would be virus wins and person dies. Could be the other way around, as in the vast majority of cases, immune system wins and virus dies. It could be something more nuanced than that. It could be immune system overreacts and lung or other parts of the body are damaged by inflammation. Or it could be a thing that lots of people have been asking about recently, and lots of journalists seem very keen on at the moment. Person gets better, but there's some kind of long-term damage, and person doesn't feel right for an awfully long time because something's gone awry and they feel unwell. So lots of possible outcomes. And lots of research for keen people to do. If any of you are keen on immunology, plenty of unknowns still up for grabs. Esther and Karim have done a good job of introducing you to the immune response. So my little reminder of that is that life on this planet is very dangerous. And if you want to be a very successful species, like a Homo sapiens, you really need to devote quite a lot of evolutionary energy and genomic energy to having this multifaceted machinery for defense, especially against nasty microbes like this one. So you'll have gathered from everything I've said so far that I don't really think protective immunity in this infection is just antibody. And as immunologists, the thing that we spend an immense amount of time debating is, in that case, what are the correlates of protection? Okay, what does it really take to be protected from this thing? Because if we don't really know what effective immunity looks like, how on earth are we going to work out who has it, how many people have it, whether enough people have it, how long it lasts, or what on earth you might want to do to mimic that kind of protective immunity in an effective vaccine? So lots of things we need to know. So, our obsession with this term correlates of protection, and because we like abbreviating things into hard to understand acronyms, we just call them COPs. So a COP, what is it? It's a level of immune response or a type of immune response that you could measure in a lab that would be considered okay to protect somebody from infection. And the COP, the kind of immunity you need, would be different whether you were talking about HIV or malaria or tuberculosis or SARS-CoV-2. But if you don't know what it takes to protect you from it, how on earth are you going to know how to design your vaccine or how to measure whether your vaccine is doing any good? So some of the more detailed protective immunity questions we really need to address as we sort of build up all the research is, again, the questions I've been asked a million times. If I've been exposed to this virus, or if I've even been sick with the virus, or maybe been PCR positive with the virus, is it a given that I've made an antibody response? Lots of the antibody tests that are around and have been debated and talked about and talked about in all those Downing Street briefings are essentially binding assays, things like ELISAs, where you attach a bit of viral protein to the plate and see how much antibody people have that can bind onto that plastic plate. But is that really the same thing as the real deal? The things I showed you in that manga comic at the beginning, the hero antibodies that could actually neutralize a virus before it gets into the cell, unknown at the moment. And the kind of thing I was alluding to just now, we speak very loosely, and there was a lot of politics going around about this at the beginning about herd immunity. But what on earth is it, and how much is enough? So I brought up the idea of antibody tests. It's been a surprisingly contentious issue. So obviously, you certainly need to have some way of measuring whether specific antibody is present or absent. And people talked a lot, didn't they, about relating this to the sort of principle of a pregnancy test kit and the idea that you might buy a little module from Amazon or from a shop that would give you a black line or a coloured line or not and say whether it was present or absent. But perhaps you need something a bit better than that, because perhaps you want to know how much antibody is present, because some people clearly make loads and some people make hardly any, and surely that might make a difference. So you might want a fancier test, and that's a point that I think we got to more recently. And the thing I was alluding to just now, perhaps you want to go one better than that and know whether people are really making the functionally meaningful antibodies that could prevent a virus getting into one of your cells. So immunologists over the last few decades have got amazingly good at making monoclonal antibodies, which you can produce in enormous quantities and characterize and analyze and make in factories and give them back to patients as a treatment to block viral infection. And there's an immense amount of quite exciting work going on that at the moment. And that'll be coming along, I think, quite soon. And then to finish off, I just want to talk a little bit about the detail of the data and what it looks like when you measure antibody responses. So there've been a vast number of papers, again, appearing virtually on a daily basis on measuring viruses in people, measuring antibodies in people, measuring different aspects of immunity, and trying to relate that to how well or badly they do. So this is a paper that's searchable on Google and available like all the others. And it's one of the early studies out of China where they attempted to do all of those correlates of how much virus you have on board and how much antibody you make. And it's an early cohort of 67 patients who were hospitalized from January to February. And they just looked at how long it took to clear the virus and how long it took to make some antibody. And I think this is one of the few bits of data I'll show. But if you scan down this table in that column IgG, which is the main kind of effector antibody that matters here, you'll see that even if you follow these people up for over a month, there's about 13% of them that never get round to making any antibody. There's well, what is it? It's 2 out of 15 patients who have clearly been infected but never made any antibody. And every study I've looked at, there seems to be a kind of tail of 10 or 20% of people who've clearly got the virus but haven't made any detectable antibody. If you look at the T cell response, the sort of take-home message, I think, is that the people who do very badly have problems in their T cell response. They're both depleted and exhausted, but the people who are going to do well have massive T cell activation. So, you can see enormous food for thought there, and lots more research that needs to be done to explore the different limbs of this immune response. So the only other point I really wanted to make about all of this is that we can learn quite a lot by looking at the other coronaviruses of the 21st century. So this is long-term follow-up of people who had MERS, the last serious coronavirus outbreak that we had. And it's helpful because people always want to know how durable the immune responses are, and obviously hard to answer for a virus that we've only known about since January the 11th. But for MERS, we've known about it much longer, and some of those patients were followed up for years. And so this is a graph where we're looking at the amount of neutralizing antibody against number of days since they were hospitalized. And I think it tells a story, doesn't it? It's kind of sort of good news and bad news story. So some people make antibody and they make it at a reasonable level and it hangs around, for a year and a half. And some people make a really rubbish immune response that sort of trundles along the baseline of the graph and never really does very well. So maybe that's a sort of warning for us if we're trying to think about variability and durability of response in these infections. So I hope that's been helpful. So the points I really wanted to bring out were that this is a new virus. We've only known about it since January. Every possible question is unknown at present, and people have been racing in labs around the world to try and fill in some of those gaps. Many possible outcomes from infection. Some people hardly know they've been infected. Some people do very badly. Some people die. Immunity to any virus is a really complex, multifaceted thing with many parts of your immune response playing its own role. And we're trying to narrate the story of what's special about immunity to this infection. And to do that, we need really reliable tests to understand how different parts of this immune response relate to the outcomes. We need to know these correlates of protection that we're so obsessed with. Otherwise, how on earth are we ever going to sort out who's protected and who isn't? And all of this information needs to be fed into generating the best possible, safest possible vaccines to try and mitigate any future involvement with this virus. So I'll stop there, and very happy to take questions now or on email or Twitter or Pigeon or any way you like.
Great. Great. So really, thank you, Danny. That was really fascinating. I think it's important to mention at this point that we will put these webinars up on our website so you can listen again, because there was a lot of information in there. So I've got lots of questions myself, but I'm going to go to the obviously go to the questions that come through on the chat. So Danny, if we just first ask some questions based on your talk. So Jake Evans has asked us, do the antigens on COVID-19 change like flu? And what are the implications of that?
Yeah, it's a really important question that lots of people have been asking. And it's a very simple answer that all viruses are quite good at mutating. Coronaviruses are fairly good at mutating. And there've been something like 24,000 different sequences around the world so far. And they're all a little bit different, but they're not amazingly different in the way that flu is. So it doesn't look like this is evolving in some kind of scary way into a worse virus or a more evasive virus. It's just, carrying on with its business. So yes, it is mutating, but not in a very scary way.
Why do they mutate? Why do they need to mutate?
Because it's a brilliant thing to do for their evolutionary advantage, to be not very good at copying your nucleic acid accurately, so that you can produce variants and profit from that in the face of selection pressure. But I think I'd argue that this has been, by accident, such a brilliantly successful virus that it isn't under any selection pressure. Humans haven't placed it under any selection pressure. It's carrying on with its own business brilliantly.
Okay. Okay, then the next question is from Rowan, and it's if you have a less severe form of COVID-19, because we obviously know some people are, getting the full works and other people have just very minor symptoms. So if you are just sort of maybe having a bit of a sore throat and a cough and it doesn't develop, do you produce less antibodies, and therefore are you likely to be more prone to infection than others that have had the really full-on severe disease?
Yeah, a brilliant question. It's really comes to the heart of the matter. So, and in fact, the perfect paper came out on this question about 3 hours ago from the Netherlands.
Hot off the press.
Yeah, and they've got brilliant graphs in it where they do all the things you'd want to see. They very precisely, they take a cohort of patients and they very precisely quantify how much virus they have on board, and then they relate that to how much neutralizing antibody they make. And the answer is terribly clear that it's an absolute linear relationship, that the more virus, until you get to death and too much virus, the more virus you have on board, the more antibody you make. And once you start to make significant amounts of antibody, you lose the virus because you've neutralized it. And so if you're one of those people who's had on board maybe 1,000-fold less virus because you've been rather mildly infected, you might be one of those people on the bottom of the graph I showed who are barely above baseline and don't have much protective immunity for next time around.
Right. Okay, so there's pros and cons. God. So I just wanted to mention that this type of research that Danny's talking about, where you're looking at patient samples and looking at responses of patients to diseases, to, for example, here we're looking at infection, and now there are and now there are trials going on where people are looking at the effect of drugs on these treatments, on these infections. That type of research is called clinical research, and very often you have people that train as doctors to do the whole sort of medical degree, and then they get interested in clinical research, and we have a lot of them in our university that work part-time in hospitals and also do the clinical research on patient samples. But also we have basic scientists doing that clinical research, and for example, Karim is one of those. So as you heard earlier, he's getting his samples from patients. So there's a couple of questions here for you, Karim. So one is from Cam, and he's asked, could there eventually be a cure for asthma? Big question.
Yeah. So we're starting to know now that asthma is actually a very complex disease, and there's many types of asthma. So you have, for example, allergic asthma, non-allergic asthma, you have steroid-resistant asthma. So the mechanisms behind each of these diseases is slightly different, and different immune cells function in different ways. And so while we're at the point now where we're starting to understand how these cells are interacting with each other and how they're leading to disease. And indeed, there are clinical studies going on looking at the effect of various therapeutic agents on asthma outcome. And so I do think the field is progressing. Whether we're at the stage where we can find a cure right now, I don't think so. But I think in in the near future, we'll definitely be seeing more and more, treatment strategies coming out and stuff like that.
Yeah, definitely. And I think the other important point is that people now know that there are lots of different types of asthma. It's a bit like cancer. Yeah. Originally, it was just called cancer. Now we know that there's leukemia and pancreatic cancer, and you might treat them in very different ways, and it's the same for asthma. So there's another big question for you. So this one comes from Akesha, and it's for Karim, and it says, is asthma genetic, or do people get it from their environment?
Okay, very good question as well. So we know that asthma is a disease that's multifactorial, and that means that it's caused by potentially many different mechanisms. And so we know that in some types of asthma, there is a larger genetic component. So if you know, some of your family have had asthma, you may be more susceptible to it because you've inherited their genes. However, there is also a lot of evidence to show that the environment does play a big effect on the development of asthma. And one of the kind of main ideas in asthma research is this idea of the hygiene hypothesis, whereby individuals who grow up in cities which are more, they have a higher level of hygiene, actually develop asthma more frequently than individuals who have grown up perhaps in more rural environments, such as, you know, on farms. And so it does look like the environment is playing an effect, and a lot of research is going in to see at which age is the most critical in determining the effect of the environment on asthma susceptibility. So yeah, it's a mixture of both, depending on the type.
Great. Okay, so thank you all. I'd like to really thank Danny, Karim and Esther for giving us an insight into the world of immunology, and in particular helping to understand our sort of body's immune response to COVID-19. We'll all be thinking about the sort of B cells and T cells if we are unfortunate enough to catch it. Thank you all for joining us in the live stream today. Everybody out there, I hope you've enjoyed it. I hope you found it informative, and really thank you for your questions. So tomorrow, if you'd like to join us, we'll be hearing from students, this time from the Department of Chemical Engineering. So why did they choose to study chem engineering? And we're also going to be hearing from a couple of students that have spent the last couple of months in the lab actually making hand sanitizer for the NHS and for care homes, and they've been making it from gin. So I'm really interested in that, and that sounds like a serious wet lab project. So until tomorrow, I'm just going to say goodbye. Thank you.